Hi, welcome everyone. Welcome to the fourth event in our Building Brain Awareness to Help Brain Injury webinar series. We are Synapse at the University of Oregon, an organization whose purpose is to serve, support, and advocate for local brain injury survivors. We help organize and foster opportunities and programs for community members as a way to combat social isolation and increase awareness for the brain injury community. We do this through peer support programs, social events, educational webinars, and symposium series such as this. We hope to create long-lasting and meaningful connections and encouragement for the brain injury community. As an advocate for individuals living with brain injury, we here at Synapse at the University of Oregon are very grateful you decided to join us today for the live presentations. We want to send a huge thank you to our wonderful guest speakers joining us today and in the coming weeks. So first we have Dr. Toby. He received his BGS from the University of Michigan and then later his PhD from the University of Washington in physiology and biophysics and psychology. Some of his areas of expertise include behavior and cognitive systems neuroscience and the neurobiology of spatial orientation and navigation. He is currently working on using single unit recording techniques to address issues related to the neurobiology of spatial cognition. And then later we have Dr. Elizabeth Powers. She finished her vision therapy residency in June of 2018. She's now an associate doctor and is assisting patients in Hillsborough. She gets to do a little bit of everything from seeing patients who have suffered from brain injury to routine pediatric eye exams to working one-on-one with amazing patients in the vision rehab department. Elizabeth earned her doctorate from Pacific University College of Optometry and received the Dean's Award and the Toll Greenstein Award for Binocular Vision and Vision Therapy in 2017. So before we get started, I would like to request that if you have a question to please put it in the chat. We will be answering those questions, or the speakers will be answering those questions after each of their presentations. So, Dr. Toby.
Okay, hi. Well, thank you very much, Emily, and a very nice introduction. And thank you, Daniel, for inviting me here.
Thank you for coming.
Sure. Do I share my screen now?
Yeah, if you can't, please let me know, but you should be able to.
Okay.
Can I say a few words just to give a little introduction? Because I'm excited about this seminar. This is one, so brain injury to our nervous system is really, really strange. And when I had my brain injury, I had injury to one of my cranial nerves. I had double vision for many months until I had, my mom found an experimental clinic that had designed this, this paradigm, and it worked for me. It took a couple of months, but I got my vision back. I think about all this stuff, like there's the thinking in our head at these levels of what's in our brain, and then there's the actual facts of what changes in you, and then there's the clinical work, and then there's the crazy amount of work that you have to do. So there's all these levels to talk about, and I'm trying to like smush them all into one place, and it's hard to do that. But I'm really excited about this talk because Dr. Taube has a very interesting perspective on the levels of specific circuits in the brain, and it really relates with what Dr. Powers is going to talk about. And I just think it's helpful when you have these kinds of injuries to have some understanding, and it also helps to frame the amount of work that needs to be done as you slowly rebuild these changes. I know there's some really cool technology out there with like the Mind Eye Institute has these awesome brain wear glasses and stuff. And I don't know where the talk is going to go fully, but I just wanted to share that we're trying to like hit all these levels, but it helps with the perspective when you're dealing with these kinds of injuries. So I'll be quiet now. Thanks.
Okay. Let's see. Oh, come on. Okay. So, can you see my title slide here?
Yes.
Okay. And you see my pointer? The red pointer?
Yes.
Okay.
I can see it. I think everyone can. Yes.
Okay. So I'm going to, so I can see my screen well. Actually, let me do this. Hold on. Well, I can't see you. So as we get going here, if you have a question, you should just jump in. Don't worry about putting it into the chat, and just ask me. I can't see you, so So make sure you just speak up whenever you can, okay? So there you go. All right, so the title of my talk here is The Role of the Vestibular System in Perceived Directional Heading. And I'll start out with this nice picture of a herd of elephants in the country of Chad in Northern Africa. And this was taken from the National Geographic, I think 12, 13 years or so ago. And this herd of elephants migrates from season to season over hundreds of miles. Usually there's a matriarch elephant sort of right here, who sort of leads the pack. And she leads them, from one watering hole and feeding ground to another. And it's remarkable if you just think about how she and the rest of the elephants do this in terms of, this is a featureless plain, taken from the perspective of, where the elephants are. There is no like Mount Kilimanjaro or something like that in the distance that they can use as a reference point. But they just, basically navigate over this entire plain, repeating this migration route every year. And you could just sort of start out by thinking about how do they do it, okay? Now, in terms of how they do it, this is one of the areas that our lab is most interested in. So our lab focuses on the neural mechanisms underlying spatial orientation and navigation. And over here is sort of a list of things that we study. We don't have to get into that. But let's start out by, when you talk about navigation and sort of the neurobiology of it, it's important to realize that the brain utilizes two fundamentally different systems. The first system, as I have on this slide, is called inertial navigation or path integration. And it tends to use internal cues in the body that basically are monitoring the different movements that your body and head are making. And you use these internal cues, let's say, to keep track of, let's say, how your head might be. So here's my head is turning now like this or moving up and down. And you might keep track of how your head is facing as you move about the environment. And it doesn't even have to have a head movement. Obviously I can just be walking. And if I just turn my body like this, even without a head turn, I'm going to be changing the direction of which my head is facing. And as I track that or keep track of how my head is facing, you're basically updating what the new head direction is as after you turn your head. So if I know that my head is, let's say, facing towards the computer screen here, and then I turn my head like this, I know I've turned my head at a certain speed for a certain angular distance, and I could sort of take that and update my current head direction is, let's say, 45 degrees, let's say, to the right, clockwise of the computer screen when I do that. Now, in order to do that effectively, you have to monitor these internal cues to the body continuously, as opposed to episodically, right? And this internal sort of inertial navigation or path integration system uses a number of different cues. They're all what we say internal to the body. So we use the vestibular system, and I'll get back to that in a little bit later. You also use proprioceptive cues, which are just feedback information coming from your muscles. And these are sometimes also called kinesthetic cues. You also use what are known as motor efference copy cues, which are sort of copies of the code that the motor system sends out to your muscles. So for instance, if I'm going to raise my arm like this, let's say up, obviously my motor cortex is sending a signal out to my spinal cord and the appropriate muscles in my arm to basically to raise it. Well, in addition to that signal that's coming from or originating in the motor cortex, there's a copy of that signal that the motor cortex might send to other places in the brain, and forming other places in the brain that, hey, I just sent a signal, down to your spinal cord to elicit the arm movement. And that's sort of that copy of the signal that gets sent to other brain areas is referred to as a motor efference copy. Okay, so these are examples of different types of cues that are used internally in this sort of inertial navigation or path integration system. The other system to think about is a landmark-based navigation system. And I don't know if any of you are sailors, but typically the terminology that sailors use is sort of taking a positional fix. And in the landmark-based navigation system, as I have here on the screen, you use familiar and very sort of salient external landmarks, cues that are outside the body, someplace in the environment, that are generally stable. You're usually large, but they don't have to be. And you use these cues in sort of an episodic fashion. I know, like, for instance, if you're living in Seattle, you might use, Mount Rainier as obviously a landmark, because you know basically where you are usually relative to Mount Rainier. Mount Rainier is south of Seattle, so that gives you a general clue in terms of what your direction might be in the environment. If you're in Portland, you might be using, Mount Hood or some other, famous building in downtown Portland. So those are examples of landmark-based navigation. Usually for landmark navigation, you use visual cues, but you could also use auditory cues. If you're living in Seattle, you might hear the distance, foghorn of a ferry boat or something like that to give you a clue as to where you might be relative, to Puget Sound. You can also use olfactory or somatosensory cues as well. You can sort of, in the dark, you can sort of touch things and feel yourself around in a particular room. And there you're using your tactile cues, to touch things in the environment. Again, it's an external cue. But to use all these external cues successfully, you have to use them, or you tend to use them in an episodic fashion, as opposed to, continuous monitoring fashion, as you do with these inertial navigational systems. So it's not like, and what I mean by episodic, it's not like you have to stare at Mount Rainier, 24/7 to know where you are relative to Mount Rainier, but you just generally refer to it in an episodic manner whenever you need to update what your spatial orientation might be. So these are the two systems that are used, and you can think about it in terms of which systems are the elephants using, as they're migrating on their routes in Chad. Okay, so let's back up a little bit and ask, what are the neural mechanisms that underlie your spatial orientation and navigation? And you can sort of start by asking, what are the building blocks in the brain to build up your sense of spatial orientation, and that allows you then to navigate from one place to another. And I should sort of say, before we get into the nitty gritty of this, spatial orientation and navigation is sort of a cognitive process that most people, take for granted. I mean, usually most people can do it fairly well, and you don't think about it really. But if the areas of the brain that are involved in these processes go awry for some reason, you obviously are going to have some immediate impairments that you're going to recognize immediately. Like, you might get lost, you might have difficulty, navigating from one place to another. Okay, so what are the building blocks in the brain? And a lot of the...
May I ask a question?
Sure.
This is Randy Miller. So I don't know if I'm sure this applies to others, but I don't know about everybody in the group here. What I'm wondering about is, everything you're speaking about relates to navigation, navigating the physical world, so to speak. What I'm wondering about is, if you have what are called social deficits, which brings up a whole different range of social cues and interactions with people, does any of that relate to the navigation that you're speaking of? I'm not even sure how well I posed the question.
Well,
Dan, do you understand what I mean?
Somewhat. I'm not sure if I can help you out here. I mean, in terms of social cues, that's like a whole another field. I mean, we're thinking more about, like wherever you are now, whether you're in your office or your house.
In the physical world.
Yeah.
Yeah. Okay. That's what I was just wondering. Okay. I'm sorry. Continue.
No,
I think
okay.
that
So
there is a commonality in the sense that the building blocks that Dr. Taube is about to discuss, there may not be the same underlying social interaction, but like the stuff Dr. Taube is about to talk about, social interaction, social cues have a whole underlying system. And as a brain injured person, I kind of got into this perspective that everything in me has that, especially the parts that were damaged or injured. So yeah, it applies, but it's just a different system. It's kind of a hand waving answer. So
Okay. All right.
Yeah.
All right. Good. Continue. I'm done.
Okay. Sure. No, I think your question is an interesting one. It's just a much different field, I guess. You know.
Yeah. That's what I was wondering. That's kind of what I was wondering about. I wasn't sure how well what you were talking about to talk about and how well it might have related to that.
Yeah.
So go ahead, continue.
Okay. So one of the most important structures in the brain that's involved in spatial orientation navigation historically has been the hippocampus. And here's a picture of a sort of a dorsal view of the brain with a cutaway view here. And this is the hippocampus right here. You also, this is on the right side of the brain. You obviously have one on the left side too. And most people think of the hippocampus as being very important for memory, and it certainly is, but we're not going to really discuss those aspects of those functional aspects of the hippocampus here, but more in terms of its relevance and importance for spatial orientation and navigation. So why do I say that the hippocampus is important? And there's basically three lines of evidence to sort of demonstrate this, each using a different approach. In the behavioral approach, which is shown in this slide, we have the infamous what's known as the Morris water maze task that rats do. And I don't know, again, if you are familiar with this or not, so I'll go over it just briefly. You know, there's a pool of water here, a large pool of water that you place a rat into. And rats, they can swim naturally, but they don't, you know, like continuously swimming. So they're naturally inclined to try to find a way to escape out of it. And what you do is you place a hidden platform like this just beneath the surface of the water that they can climb up onto, but they can't see it because the water is made sort of murky or cloudy by adding some powder or milk to it or something like that. So they have to learn to swim around and basically find where this hidden platform is in the pool. And once they find it, you know, on the first trial, it's usually just by accident. I mean, of course, because there's no way of knowing that there's a platform there, but eventually they stumble across it. And once you give them several trials at it, and the platform is always in the same location in the pool within the room. And so after several trials, they quickly, wherever you release them on the periphery of this pool, they'll quickly swim a direct line basically to this platform and climb out. So before they know where the platform is, a typical route might look like this red line, but then once they figured out where a platform is, they swim directly to it no matter where you release them from. And I should say, if you make a lesion in the hippocampus, rats don't do this, very accurately. They look more like the rat here on the left, just swimming all around. So they can't really learn to figure out where to find the platform, at least animals with hippocampal lesions. A second approach, yet a different approach, was shown very elegantly, now over 20 years ago by Eleanor McGuire, who works in London. And this was an brain imaging study where they took taxi cab drivers in London. And I don't know if you've ever been to London, but if you're a taxi cab driver there, you have to have you have to take a test actually to be a taxi cab driver there. Because if you've ever been to London, and here's a sort of a really small map of London here, but London is not laid out like a grid like New York City is, but rather there's lots of curvy streets, lots of angled streets, and it's sort of basically a mess. And yet the taxi cab drivers obviously have to find their way through this in order to, bring their passengers to whatever goal their goal happens to be. So what Eleanor McGuire did is she took these taxi cab drivers and she put them in a brain imaging unit, and she basically imaged their brains while they were asked to sort of describe a route. Let's say it's a little bit hard for me to see this here. Maybe if, let's say from, Park Lane here going to the Elephant and Castle place down here in the lower right-hand corner in London. And they, these taxi cab drivers would basically have to, explain the route that they are driving in order to get here. Sometimes they might have to go by, Buckingham Palace or something like that. So while these taxi cab drivers were explaining the route, Eleanor Maguire and her colleagues imaged the brain. And what they found is the area of the brain that lit up, that was most activated, right, is shown here in yellow, which is basically the right hippocampus. So again, another study showing that the hippocampus is important for basically describing these routes. And the third study, again, yet a different approach that shows that the hippocampus is important for spatial orientation and navigation, comes from physiological studies that were initially done by John O'Keefe back in the early 1970s, of which he later won a Nobel Prize for about 5 years ago. And O'Keefe and his colleagues showed that if you were to record from individual cells in the hippocampus, that one cell would fire when the animal was in a particular place. And so this is a overhead view here of a cylindrical apparatus. Maybe it's about, I don't know, a meter and a half across or so. And the animal just runs around here in the cylinder, maybe collecting some food pellets that you throw in arbitrarily. And this particular cell that they were recording from happened to fire where you see these dark blue squares. And this is basically a color-coded firing rate function map that just shows the firing rate of the cell based on the animal's location. And basically the darker the color, the higher the firing rate. So blue is the highest, green, then light blue, then green, then red, etc. And all the yellow squares are basically places that the animal visited, but the cell did not fire at all. So as you can see here, this cell liked to fire in the upper right-hand corner. This is just one cell. And if he recorded from a different cell, he would see in the hippocampus, he would see that a different cell would do the same thing, but maybe it would fire maybe in the lower left, and a third cell might fire in the lower right, and the fourth cell might fire in the center. And if you looked at the entire population of the cells, they basically mapped out the entire environment that the animal was locomoting in. These cells came to be known as place cells, and each individual place cell basically fired based on a particular location that the rat was in the environment. Again, these were in the hippocampus. And then if you were to look at the structure that sends its most prominent projection to the hippocampus is known as the entorhinal cortex, or EC, and it provides the major input into the hippocampus. And cells in this area sort of fired sort of also based on place, but rather than having a single place, they had multiple places that they'd like to fire. And these multiple places basically mapped into sort of a grid-like pattern, as is shown here on the lower right. And this became known as what's called grid cells. These were discovered around 2005 or so in the entorhinal cortex. And although they're not firing in a single place, this multiple, spatial pattern is obviously very spatial. And this is the major input into the hippocampus. So the hippocampus is receiving some major information about spatial aspects of the animal in the environment. So like with a grid cell, for instance, it would fire at all, one single grid cell would fire at all these places here, shown in yellow and red, but when it was in a blue spot, it would not fire at all. Okay. So again, just summing up, basically what you can conclude is that the hippocampus is important, or certainly very involved in spatial aspects of the animal's environment. All right, now if you think about if you're going to navigate, let's say from, I don't know, your home to your office, even before you think about the route you're going to take, the first thing you have to ask yourself, even before you get up from your chair, is you have to ask yourself, what is your spatial orientation in the environment? And there are two aspects that are involved in your spatial orientation. One is your location. Where are you in the environment? And presumably that information is encoded by place cells that I just talked about. And the other information that's very critical is what direction are you facing? And the information that place cells are providing about location really don't inform you about what direction that you happen to be facing. So here's, again, a map of a place cell that like to fire in the lower left here. And it wouldn't matter if the animal was facing up or down or left or right, the cell would always fire whenever the animal was in that place. So there was really, these place cells were not providing any information about what the directional heading of the rat might be. And obviously what your directional heading is going to be very important for being able to navigate. So in order to provide information about your directional heading, you're going to need another type of cell. And this other second type of cell became known as head direction cells. These were discovered in 1984 by James Ronk, who's pictured here, who actually happened to be my postdoc advisor way back then. So to give you an idea of what a head direction cell looks like, I'm going to play you a little video here. Hopefully this works well. And let me just turn up my volume here. So what you're going to see here in this video is you're going to see, again, this is a top-down view of the cylinder with a rat that's going to be running around looking for little food pellets. And as it's running around, you're going to hear the cell. And what I want you to do is listen to when the cell fires and where the rat is, as well as what direction its head happens to be pointing. And I'll give it away right away so that you can look at this as I play the video. And that is, this cell is going to fire whenever the rat looks sort of towards the lower left at about 8 o'clock, right? And it won't matter where the rat is in the environment, but it'll always fire whenever the rat looks at 8 o'clock. So the cell hasn't fired yet. The rat's just looking for food pellets that are thrown in there randomly. And right there, she faces 8 o'clock. And if you watch carefully, every time she faces 8 o'clock, this cell will fire, independent of where she is. There, 8 o'clock. Eight o'clock again. Now, if you watch carefully, she's going to come up here and she's going to face 9 o'clock, which is a little bit off of the 8 o'clock. And this, you'll hear the cell fire again. It's going to fire at a lower firing rate. Right here, I think. That was 9 o'clock and it fired at a lower firing rate. Contrast that to 8 o'clock, right? Which it fired at a much higher rate. So this is a head direction cell. And you can see that this cell fired whenever she was facing 8 o'clock, and the firing rate fell off as her head came out of the angle, at 8 o'clock. Again, this is one cell. If you looked at another cell in this area that might be nearby, you would see that this other cell would fire similarly in a head direction like manner, but maybe you'd be tuned to 11 o'clock. A third cell might be tuned to 3 o'clock. And if you looked at the population of cells in this particular brain area, you would see that they were pretty much uniformly distributed over 360 degrees. So in other words, you could find cells tuned to 50 degrees, cells tuned to 100 degrees, cells tuned to 180, etc. Right? And each cell was tuned to a particular head direction for the rat. So that's a head direction cell. And this slide here just sort of sums up some of the properties. It's the direction of the head, not the body position. It's the animal's head direction in the horizontal plane. It fires whether the animal is moving or is still. And its firing is independent of the location that the rat is at in the environment, as well as its ongoing behavior. Each cell only exhibits one preferred firing direction. And as I mentioned, the preferred firing directions across all the different cells are pretty much equally distributed around 360 degrees. And you can plot tuning curves for these cells, such as I have here, that plots the firing rate here on the y-axis as a function of the animal's head direction from 0 to 360 degrees on the x-axis. And this is sort of the tuning curve of a typical cell here. It has its preferred firing direction at about, this one at about 180 degrees. It has a peak firing rate of about 20 hertz, 20 spikes per second. And then this is the directional firing range, which is basically the range of head directions that are firing above that of the background level. Background level in the video you just saw was essentially 0. The background rate for this cell, was down around 1 spike or less per second. And just to make sure everyone's with me, if you were to draw a vector of where the rat is and what direction its head was pointing whenever the cell was firing, you would see that all the vectors basically are parallel. So all these vectors point at 8 o'clock. It's not like they all converge to a single point, here on the circle, but rather they are all parallel with one another. So that's a head direction cell. Okay, here's a little bit brighter picture in the cylinder of this rat running around. And one of the typical questions I should mention as you're looking at this picture, that we purposely provide the animal with a very salient landmark cue, which is this white piece of cardboard that's taped to the inside wall. Surrounding the cylinder is a black curtain that you can see here in the background that goes from the floor all the way up to the ceiling. But the environment itself is pretty uniform, and it's only really broken up by this white piece of cardboard that's attached to the cylinder. So people often ask, well, what happens if does this cue card exert stimulus control over head direction cell tuning? And you can look at that or answer that question by basically rotating the white card. So the card starts out, so here you have a head direction cell tuning curve shown in black here in the first session with the card positioned at 3 o'clock here. And then you can take the rat out of the environment, you put it in an opaque box, and while the animal is situated in the box, you rotate the cue card, that white card, over to the opposite side of the arena over here, right? Then you sort of take the rat that's in the opaque box and you disorient the rat by sort of turning the box around a little bit and translating the box in the room. Then you take the rat back out of the box and you put it back down into the arena and record it again. And lo and behold, if you rotated the card by 180 degrees here, you can see here that the cell also rotates by 180 degrees, meaning that the cell is sort of locked in its relationship to the cue card. And then, of course, if you bring the card back to the original position, the cell returns to its original position. So these cells are locked to the external landmarks in the environment. In this case, it's the cue card. Now you could say, well, this is just a visual cell that's responding to the cue card, but that's not the case. So in this experiment, what we did is we basically removed the salient visual cue and looked to see how the cell responded. So in the standard session here, again, we're recording with the card here at the 3 o'clock position. In this case, we're recording from 2 different cells simultaneously, the blue cell and the red cell. And you can see they each have a different preferred firing direction. The blue one fires at about 45 degrees or so. The red one fires at about 120, all right? And then we take the animal out of the cylinder again, put it in an opaque box, and we, in between the sessions, we take out the cue card, we bring the animal back in, we disorient the animal again. And when the animal is placed back in the cylinder, this is the response you get. So then now there's no cue card in here to, for the animal to use as its reference point, but yet these cells still fire in a head direction cell-like manner. Here's the blue cell here, and here's the red cell. And you can see that although, the blue cell has shifted its preferred firing direction over here, and the red cell shifted it over here, you can see that they maintain the relationship to one another, even in this sort of now cueless environment. I don't know why, how the animal decided, or the cell decided now to fire in this direction compared to what it was before. But whatever cues it was using to shift its orientation over to this new direction. You know, the red cell, the same thing happened in the red cell, such that the two cells remained sort of in register with one another. And if I were to record from 10 cells, you would see that all 10 cells basically shifted the same amount. So all the cells in this network are basically staying in register with one another. So the whole network basically shifts. Okay, so, and again, these cells don't need a prominent landmark in order for them to fire this way. They can, you know, they fire this way. It's more of a cognitive representation of the animal's perceived directional heading within the environment. Okay, so let's look a little bit about where these cells are in the brain. Again, you probably aren't too familiar with this view of the hip, but this is the hippocampus, sort of a cutaway view, a cross section through the hippocampus, that I'm circling here. Down here is the entorhinal cortex, and here's a cell here in the entorhinal cortex that's projecting into the hippocampus, which I said was sort of the major gateway into the hippocampus. And in between the hippocampus here and the entorhinal cortex down here are a bunch of adjacent brain areas, S, pre, and par, that stand for subiculum, precebiculum, and parasubiculum. And these are all areas that are independent of the hippocampus, as well as the entorhinal cortex. And these areas are situated in between the two, and they have their own sort of identity. And it turns out that these head direction cells were discovered down here in these deep layers here to the precebiculum. So pre stands for precebiculum, par stands for parasubiculum, and S stands for sort of subiculum. But these cells were basically discovered here in the deep layers of the precebiculum. Okay. Now, since that time, these cells have been identified in a number of brain areas, and we don't have to go through these, but I just want you to appreciate that there's lots of areas in the cortex as well as in subcortical areas where you can find head direction cells like this. Here's an example of tuning curves from 3 different cells. So here's a cell in the postsubiculum. Here's a tuning curve from a cell that was in the anterior dorsal thalamus. And then actually the video I showed you was from a cell here in the anterior dorsal thalamus. And here's a tuning curve from a cell in the lateral mammillary nuclei. Right? So they all look sort of, I sort of showed you different cells with different peak firing rates. So for instance, this cell here in the lateral mammillary nuclei had a very high peak firing rate, 120 spikes per second, while this one here in the postsubiculum was only firing at 16 spikes per second. But I could find cells in the postsubiculum, you know, that had firing rates upwards, you know, of 100 spikes per second. And you can find cells in the lateral mammillary nuclei that might only, you know, peak out or top out at 20 spikes per second. So there's different properties for these cells in all these different brain areas. Okay. So what is the role of the vestibular system in the head direction signal? Now, if you think about the vestibular system, it's an ideal candidate for updating your perceived directional heading within the environment. And that's because if you start with a known directional heading, like if you start out thinking, okay, you know, my head is, you know, facing again towards the computer screen, and then you turn your head, and as you turn your head, let's say clockwise, let's say 45 degrees to the right, your vestibular system can detect the turn of your head. And if the vestibular system is detecting the turn of the head, it could send that information on to the head direction cell network saying, hey, my head, you know, is currently facing, you know, towards the computer screen, but now the vestibular system says, I just turn my head 45 degrees clockwise. And so you can sort of compute what your resultant head direction is going to be after that head turn. So the vestibular system, which is sort of shown here in this slide, is an ideal candidate for updating what your current perceived directional heading might be. So again, I don't know how much you know or are familiar with the vestibular system, but this is a diagram of it here. Here is, sometimes people call it the inner ear because it's adjacent basically to the cochlea, which is right here. It's obviously going to be important for your sense of hearing. Here's the auditory nerve that goes to the ears. And then there's a component portion of the auditory nerve that's referred to as the vestibular nerve, which innervates this apparatus over here. And this apparatus over here is basically surrounded entirely by bone. So you have to imagine this whole thing, wherever you see white here, is actually bone. And the vestibular system is carved out of this bone. So sometimes that's also, the vestibular system is sometimes known as the bony labyrinth, because basically it's carved out of bone. And within the carved out portion, you have these membranes that form two components basically to the vestibular system. And these two components are known as the semicircular canals, right? So here's the semicircular canals. And you have three of them. You have an anterior one. Well, here's the superior one here. Here's a lateral one right here. And here's a posterior one right here. And they basically exist in the three different planes in 3D space. So you've got one like this, you got one like this, and you got one that's going to be facing the screen like that. So each of these semicircular canals are situated in a different three-dimensional plane, right? And associated with the semicircular canals, oh, I should mention, let me just go to the next slide for a second. The semicircular canals serve one function and one function only, and that is to sense the angular acceleration of the head, right? And acceleration, if you remember from maybe high school physics, is nothing more than a change in velocity. So these canals, which are again situated adjacent, pretty close to your ears, are going to be sensing how fast or how quickly you're accelerating your head, let's say to the right or to the left, all right? It turns out that the canals themselves, or the sensory epithelium in the canals, are sensitive to acceleration, but the signal that gets output from the sensory epithelium to what's known as the vestibular nuclei is actually not an acceleration signal, but really a velocity signal, how fast you're turning your head as opposed to how fast you're actually accelerating it. So the information that these canals are sending to the brain are basically is an angular velocity signal, okay? And then the other,
Toby, sorry,
yeah.
I hate to rush you. Just for the next speaker to have enough time, if we could
Sure.
just maybe have like 5 or 6 minutes, and this is perfect for segueing into her talk too. If you have
Okay.
any info on the brain levels that receive this, but if not, no worries, because but yeah, just I hate to do this,
Yeah,
but
no, I know. I'll let me just sort of sum up here, like 5 more minutes.
yeah, that sounds good. Thanks.
Okay. The other portion of the vestibular system are known as the otolith organs, and they're composed of the utricle and the saccule, and that's these two bulges that you see right here. And the utricle and the saccule are important also for one thing only, and that is to sense not angular acceleration, but rather to sense linear acceleration of the head, how fast your head is moving like this, or maybe left and right like that, or maybe up and down like that, okay? So that's linear acceleration, and that's what the otoliths are important for. Now, if you think about gravity, gravity is also defined by an accelerative force. So the otolith organs, they are sensing linear acceleration, but because gravity is basically considered a form of linear acceleration, basically the otolith organs are also important for detecting where gravity is from. So just as you're sitting there now in front of your computer screen, there's a signal coming from the otolith organs that's going to the brain that's telling you where gravity is coming from, basically, down like that. And of course, if you tilt your head, it's going to tell you that gravity is now coming, not from your, let's say the center portion of your neck like this, but rather it's coming, from the side of your head. So if you tilt your head in any particular way, the otolith organs are going to be telling you where gravity is. Okay, now how does the head direction cells interact with the vestibular system? And just to show you how important the vestibular system is for the head direction cells. Here we have a head direction cell, actually two head direction cells, these black lines that we were recording. And then we made a lesion, a permanent neurotoxic lesion, again, of the vestibular system. And by injecting a neurotoxin basically into the inner ear. And 1 hour after the injection, you can see that the cells are still there, the blue line. 24 hours later, the red line, you can see that this cell is gone. This cell is sort of on its way out. And I didn't show you a line for 48 hours, but at 48 hours, and certainly by 96 hours, the green line, you can see that these cells, the two head direction cells, are no longer providing information about the animal's directional heading. So make a lesion in the vestibular system, and it basically disrupts this head direction cell signal. Over here, we have a temporary inactivation of the vestibular system by injecting tetrodotoxin, which is a sodium channel blocker, into the inner ear, which basically blocks the signals going from the vestibular system into the brainstem. And here, if you did that again, here's the nice cell. And then after injecting the tetrodotoxin, you can see 1 hour, 12 hours, 24 hours later, the cell is not providing any information about directional heading. But after 48 hours, the cell comes back, right? So again, you knock out the vestibular system and you're going to knock out information from these cells. And I think, yeah, let me see here. Maybe I'll just end here a little bit in terms of how, just for that you appreciate a little bit how this head direction cell signal is dependent upon the vestibular system in terms of how the circuitry goes from the vestibular nuclei down here up to the thalamus. There's sort of two different pathways in the brain. There's a posterior pathway and an anterior pathway. And it's this anterior pathway that's thought to be involved in the head direction cell network. And then last slide I'll show you here is sort of just a summary diagram. I'm not going to go through, but again, if you just start down here at the bottom with vestibular nuclei, we know from anatomical and physiological studies that there's a projection from the vestibular nuclei going through the brainstem to a number of these structures. Here's the postsubiculum or dorsal presubiculum where the head direction cells were initially discovered. And here's where that head direction cell from the video I showed you. And you can see that these areas also project up to cortical areas, as well as the entorhinal cortex and hippocampus. So this information gets conveyed to portions of the limbic system that are very important for navigation and orientation. So I think, let me just stop there.
Thank you, Dr. Taube. Yeah, I think that last figure that you just had is really informative, because technically injury to any of those parts or multiple parts, it affects the whole system. And, we're left with these crazy problems that often leads people to people like our next speaker. Or, there's more issues too, but so yeah, thank you very much for that presentation. I think it's really grounding and showing, just the head direction part is fascinating.
Can I, Dr. Taube, could you, if you don't mind, post your email address in the chat?
Sure.
There's somebody in Colorado I think I should refer you to.
Okay.
I'll do it, if you could post your email in chat, that would be helpful.
And if there are no more questions, and also you guys can put the questions in the chat, I want to give Dr. Powers plenty of time to talk and show her next presentation, which is very connected with this. Thank you, Dr. Taube. I think that was great.
Thank you.
Yeah. So I don't know how to switch speakers.
Right now?
Hi. Yeah. I think I don't want to have you rushed. So yeah, we got till 1:30-ish, but
Okay.
cool.
We'll
Great. Yeah, that was really good. Thanks.
All right.
Bye all.
Thank
I'll
you.
stick around for a little bit.
Let's see here. Is that sharing right now? Let's see.
I'm not seeing it. Oh.
How about there?
Yes.
Okay, great. Well, hello, I'm Elizabeth Powers. I am not a researcher, I'm a clinician, so I have a different approach and kind of a different perspective than Dr. Taube. That was really helpful. My talk will be more about the visual system and some disconnect with the vestibular system, and kind of how those two need to communicate for us to know where we are in space. So mine will be coming more from the visual side of things. And see if I can make this move forward. There you go. So the two things we're going to kind of look at today are, we're going to talk a little bit about what a normally functioning visual system looks like and components of that, and then those same areas and how they're affected by brain injury, concussion, and what that feels and looks like. So kind of targeted to the functional side of things. I'm very biased, so I think vision is really important, but it can comprise up to 70% of our sensory input. Changes how we see the world and has a lot of connections to other systems in the brain and body, including those listed here, many of which Dr. Taube just referenced and discussed. So it has a lot of kind of touch points to other systems that needs to communicate with. So, a functioning visual system. Here's a just brief anatomy lesson. On the left is kind of a cross section of the eye itself. The kind of orange sheen on the inside is the retina. It's called the sensory retina or the neural retina because it's actually formed from the same type of tissue in utero that the brain and the spinal cord are made from. Those are the only three things in the body that are formed from this early tissue. So we kind of think of it as part of the brain, even though it's outside of the brain itself. It has so many connections and it's made of neurons. There are a few muscles involved that sometimes get confused with the muscles around the eye. So the green arrows there on the left are pointing to the ciliary muscle, which controls how the eye focuses the light. That kind of blob in the middle. I don't know if you can see my mouse here. This is the lens, the crystalline lens, and that shines light, that focuses light coming from outside the eye onto the retina here. And so this muscle can adapt and change. It can kind of, it's a gradient that can focus the light. And that is neurologically connected in the midbrain to the muscles on the right here that control the movement of the eyes, especially, you see here, this is too small to read. It says medial rectus. That is really what's responsible for convergence. And these two systems, the focusing on the left and the eye movement on the right are tied together. And it's very important that they're tied together. And we'll get into that, why that is in a little bit. So here's the kind of areas of vision that we experience on a daily basis that I look at in exams. Basic one that we think of when we think of vision is visual acuity, ability to resolve images, being able to see things clearly. It has to do with the rods and cones in the eye and the retina, and it's that 20/20 or not 20/20 vision in the very center. Basic stuff. There's your eye chart. But it's not all about seeing 20/20. There's a lot more to vision. So one of the things I already mentioned was the eye focusing by that lens. This is what we call accommodation. It's that muscle changes tone, and when it does, it changes the way that the eye focuses light, so we can switch from near to far. So if you're sitting at a computer right now, and you're looking at the screen, everything behind it should be pretty blurry. And the farther away it is from the screen, the blurrier it will be. Now, if you shift your focus to something across the room, now that should be in focus, and you'll notice maybe your screen is blurry. And so that's because that accommodation, it's stimulated by blur. And so the eye does these micro adjustments to say, okay, what makes it clearer, this or this? And once it finds that position, the muscle kind of locks in and you find that clarity spot. So convergence is closely tied to accommodation. This is the ability to keep both eyes pointed at the same point in space and time. So if it doesn't make you uncomfortable to do this, you can hold a finger up in front of your face like this. If you look at your finger, there should be 2 computers behind you. If you look at the computer, there should be 2 fingers in front of you. And that is because the eyes cannot point at 2 places spatially, like on the z-axis in distance. Can't point at 2 places at the same time. There's a little bit of a range where we can kind of keep things single, but from other processes. But in general, whatever you're not looking at should be double. And that's because convergence is keeping the eyes pointed on the same place on the z-axis when it's functioning properly. And this is very important, as you can imagine, for depth perception, because it's kind of a triangulation system. So if you think of on the left there, the picture, if your left eye is point A and your right eye is point B, your brain is doing these calculations all the time to figure out how far away that ship would be, what that D is in the middle. And that is kind of how we're making these constant calculations about our depth and how far we are from things in our visual world. And that's 2-eye depth perception. There are some 1-eye depth perception things that we use, but specifically 2-eye depth perception is much more accurate and gives you more rich information about where you are in space. So convergence has to do with the muscles around the eye, like in that slide I showed before, whereas accommodation, eye focusing, has to do with the muscle inside the eye, and they are connected in many ways, so that when you converge, or excuse me, when you accommodate, when you focus your eyes, you automatically converge. And when you converge your eyes, you automatically accommodate a little bit. So again, if we're doing this activity where I'm looking at my finger, not only are there 2 computer screens behind me, but they're blurry. When I shift my gaze to my computer, now I have 2 fingers in front of me and they're blurry. And so those 2 systems working in tandem with each other helps keep everything clear and single in an ideal world. Then there's tracking. These are all in different parts of the brain, I should say. So I didn't really get too deep into the neuroanatomy of this. Maybe next time we could go into that. But there are kind of 3 main areas of visual tracking that we assess and that are kind of recognized. That would be fixation, being able to keep your eyes in one place without moving and bouncing around. Smooth pursuits, so following a slower moving target. And quick saccades, which have to do more with reading. When we are reading, our eyes aren't smoothly going across the page. They're making these quick ballistic eye movements. Peripheral vision is another big area because, no pun intended, but it's most of the retina. So we talked about visual acuity being that tiny point in the middle where we can see 20/20. The rest is all your peripheral vision, and that is incredibly important for your sense of where you are in space, writing mechanisms. This is what integrates with your vestibular system, your proprioception, your reflexes, and it's very crucial, as you can imagine then, for balance and walking. So if you're walking around, let's say you're walking down the sidewalk and you're looking straight ahead, your peripheral vision is taken care of. Okay, the sidewalk has a little bump to it, has a curve. Oh, we're turning a little bit. Your visual system, not your 20/20 vision, but your peripheral vision is taking in that information all the time, trying to tell the rest of your systems how to respond and integrate with those systems. Spatial awareness kind of goes along with that, but this is also has to do with your sense of center, what we call like your egocentric midline. So there's kind of a writing principle that once you have kind of a sense of the center of your body, and this depends heavily on your peripheral vision. If you don't have good interaction between both sides of your body, whether it's from an injury or a stroke, that sense of your egocentric midline can shift, and then that therefore shifts kind of your reference point for your visual, vestibular, proprioceptive systems. So this is really important for body coordination and matched body strength and integration of both sides of the body, so you can do different things at different times with both sides of your body, is really important for maintaining balance and coordination as well. Then, another thing you think of when you think of the eye doctor is glasses, right? This is oddly enough a pretty small part of what I'm looking at. It's important getting the eyes to a really relaxed and clear state, but this is just one little piece of the puzzle. So refractive status has to do with how the eyes refract. If it's nearsighted, farsighted, there's astigmatism, that kind of thing. This is not really, there are some changes to refractive status with brain injury, but it's rarely affected too much. It's more has to do with the muscles that control the focusing rather than this, which has more to do with the length of the eyeball itself. So this is more of a structural thing that doesn't usually get affected too much by brain injury. It's your ability to focus and shift focus that's more affected. And those are two totally different things. Seems like they'd be the same. And we can treat some eye focusing issues with glasses, but this is more just what's your prescription? Are you nearsighted, farsighted? That stays pretty consistent most of your life. I put it last because it's a small piece of what we do. So that's kind of an overview of some of the big main areas. There's a lot of more finer details and nuances that we look at than that, but those are kind of the big main areas of visual function that we look at and assess and are looking for changes from in brain injury. So like I said, my perspective is more clinical, and I'm going to kind of go through each of those areas, how they are affected by brain injury, what that would feel like, what those symptoms are, and how we assess it. And then we'll do a tiny bit of treatment on the visual vestibular stuff. So accommodation is usually quite affected in a concussion. Now, if you are past the age of about 45 or 50, accommodation was already going to be going down. That's something that starting at age 14, we have a reduced ability to focus. It's because that lens, that blob we were talking about at the beginning, gets stiff, and so it can't focus as well. And so that will already be decreasing. But if you're before the age of 45 or 50, or if you're there with a concussion and all of a sudden things got blurry quickly, kind of dropped off the plateau, that can be accommodative dysfunction. So it's typically, this relationship between the distance you're looking at and the way that your eyes focus is disrupted. So there's this neuromuscular relationship between the perceived clarity, the ability to feed back that into your visual cortex and process that information that it's blurry and not clear. Then that information coming forward to the midbrain, which is kind of right in the middle, the lizard brain, and saying, okay, it's time to focus. And finding that sweet spot all happens within about a half a second. And that there's a few different places, like Daniel was saying with the vestibular, that can be disrupted and in this process. It's often that feedback, somewhere along that feedback, not the muscle being damaged, but the understanding that neuromuscular relationship between what you see and then how to enact that muscular change is disrupted. I said most of that. It can't sustain the constriction either. So a lot of times there can be an autonomic nervous system component to this as well. Autonomic being the kind of, not your central nervous system, but the kind of fight or flight. You think of autonomic, the rest and digest. This is this kind of dual-sided system that does a lot of the underlying homeostasis, the balance keeping of your body's systems. When there's a really high fight or flight tone, that's your sympathetic system, it actually inhibits your accommodation. So you can get a really poor accommodative response just because your fight or flight is really high. Because this is, accommodation is mediated by the parasympathetic system, the opposite system. So those two are always kind of in balance, but if the fight or flight is high, you'll have a low accommodation. How this usually presents is you'll get headaches up or above the eyes, typically around the eyes. A lot of people experience it behind the eyes, even though it's kind of in the eye, it's this ache. Blur when shifting your focus from near to far or far to near. Sometimes that wears off and it's just delayed. Sometimes it just never clears up. And a big one for people is if they're trying to read something for 10, 15 minutes and then they look up, everything's blurry because that muscle then isn't relaxing. You can't defocus. So it's not just about keeping things clear up close. It's also about knowing the eye knowing when to defocus, and that gets disrupted as well. Also, avoidance of a lot of reading, which includes things you need to do and things you want to do. Blur after a few minutes of trying to read, so that muscle fatigues. And it takes increased neural input to keep it sustained, and it just, there's only so much, it's kind of the theory, there's only so much energy to give. There's going to be a point where that kind of taps out. And increased fatigue after doing a visually heavy task like reading or sewing or doing something small and tedious. That can actually affect your body fatigue too, trying to use other muscles to recruit to try to help with the accommodation. So that's it. Oh yes, and we assess this in a phoropter, which is on the right. They've probably recognized this from eye exams. There's a lot of really cool stuff that we can do with this. That's not just, is it better 1 or 2? Has a lot of power for using it for assessing these things, and we can use different lenses to assess your accommodation. Convergence insufficiencies. This is probably one of the most common visual issues I see after concussion. So we talked about convergence being the ability to keep your eyes in the same place at the same time. The insufficiency means either you can do it for only a short period of time or you can't do it at all. Sometimes you'll see that someone has like an eye turned out, drifting out. I see that frequently after concussion, but it's less than half the time. And so, that's an obvious one where you're like, oh, I look in the mirror and I see my eyes aren't pointing in the same place. But you can have convergence insufficiency and still have the eyes pointed in the same place. It just can't sustain that. What usually looks like double vision, rubbing eyes or squinting. You have to close or cover one eye to read to reduce that strain in the system from trying to keep those eyes in the same place. You can get pain and aches between and around your eyes. And then of course the depth perception can be very affected. If you're not using both eyes at the same time, it can give you really poor information about how far away things are from you in space. So, Dr. Toby was talking about the optic flow and some visual inputs that help orient you with all these other, all these other systems. And this would be one where that input is broken. We'll get into that in a couple minutes. And we assess this in the office, still with the phoropter, with lots of other toys and specialty instruments. And essentially we're trying to look at how much stress that system can handle before it breaks. I know that sounds really uncomfortable, which kind of is, but what we're looking for is the maximum range of what the vision can do when it comes to convergence. Eye movement dysfunction is another big area. So the tracking that we talked about, so the muscles around the eyes become kind of uncoordinated, just like when you're, if you've had a concussion and you try to walk and you feel like every step just feels uncoordinated. There's a lot of things that are off. The calibration is off. That can happen with eye movements. So you get slow, inaccurate, or even painful eye movements, which really reduces how quickly and efficiently you can gather information. Because when you think about reading, you're reading, multiple words at a time and trying to take in that information. I have a video in a moment, and let me skip to that, of what this looks like. On the left is a dysfunctional reader. So you can see how they're kind of jumping around, pausing, going back, like we call those regressions. And on the right is just an efficient reader. This is actual infrared eye movement data. So you can see they're just taking in the information on the right, boom, 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 moving on. Don't even have to really go to each letter and each word. On the left, look how much longer it takes. This is actual recording of dysfunctional reader eyes. So skipping lines, going back. So this is kind of what that looks like in very frequently for someone with a brain injury, because number one, the eye movements are inaccurate. Number two, peripheral vision is affected. And when you don't have peripheral vision working for you, you can't judge where your next eye movement's going to be. So you just kind of toss your eyes to the next place, and then you make corrections. So you actually have to have really pretty good peripheral vision to make good eye movements. This looks like losing place when you're trying to read, feeling like the eyes aren't tracking with the head movement. So this can be very vestibular as well. Don't want to, conflate them too much, but if you the eye movements aren't moving with the head, there can be a calibration off there, which we kind of group into this. Poor reading comprehension, rereading passages, rereading words, rereading paragraphs. That's how this often looks in normal life. And it's assessed using, there's a few different cool technologies out there now for assessing how the eyes are moving during reading. We look at that in a lot of different ways, not just reading words, but reading numbers, looking at pictures, and then adding in comprehension, taking out comprehension, adding in saying words aloud versus to yourself. So there's a lot of different ways that we kind of look at that and see what slows down the eye movements. On the bottom here is someone that was trying to track that circle. There was like the black dotted line there. They were trying to keep their eyes on that dotted line. And you can see the charts of the right and the left eye, how inaccurate they can be, because there's so much slip in the system. They're not able to keep their eye in one place or track smoothly. So that's kind of what we see. This is the big fun one. Visual spatial midline shift is kind of a overall descriptor for talking about a lot of different systems that don't integrate well with the visual system. So a visual spatial midline shift I see frequently, probably 90% of people after concussion, when we measure for it, find it. This means that the internal map of where you perceive yourself to be in space is not right. And it could be off by just like a degree or two, and it's going to throw off all the calibrations. So we all make this map of our world, which includes depth perception. It includes, a lot of 3D aspects. And if you look around the room where you are right now, you kind of have a sense of where things are, even if you're sitting still. And then if you add motion to that, it becomes more complex. But we have these internal maps or we wouldn't be able to move through space. And so when we have these internal maps and the visual component is inaccurate, then all of a sudden, we have a mismatch between what we see and what we feel. And that is what can cause a lot of symptoms we'll get into in a second. So fortunately or unfortunately, the visual system, there's been many studies that show the visual system kind of overrides other inputs. So they've done studies. I thought it would be kind of nausea producing to show the video, but it's essentially like a big black and white rotating room. And they start out, they have people walk through this room, and there's some steps and there's railings, and it's like a big tunnel, a big tube, and the black and white stripes are still. And these people walk through, they learn it, they can kind of play around, get a feel for it. Okay, this is great, I know where to walk here. Then they walk through it again, same people, with their eyes closed, get through it just fine. The third time they walk through, the black and white stripes start to rotate. So they're getting this visual stimulus that the world is shifting, and all of a sudden they just fall over. They can't walk. They've just walked it. They have the muscle memory. They have good functioning vestibular systems. Their proprioception, where their feet and legs are telling them they are, is all exactly the same. But because that visual world is tilting, they just go over. And so it's kind of, if you think about it like that, that's a dramatic version, but that is how much we rely on our visual system to tell us where we are and tell us about the world around us that we can't reach out and touch. When we have this mismatch between these systems, it can produce a lot of dizziness. It also just gives us, like Dr. Taube was talking about, this continuous monitoring. That can be very fatiguing because you're continually monitoring and making these recalibrations. There are a lot of vestibular disorders that can be caused by concussion, or it can have concurrently with concussion. One example would be PPPD. It's kind of this vestibular dysfunction that causes dizziness. And we know that and a lot of other vestibular issues, vestibular visual issues, there's a slowing of the communication between the visual system and the vestibular system. And that can go both ways. But I have this illustration here. So on the top, we see on the left is vision, on the right is vestibular. So what this is representing is the vestibular system ordered some blue balloons, okay? And the visual system says, all right, we got that, and sends it. And it goes fast, gets the information, the vestibular system talks back to the visual system, and there's this coherence there. So that truck just moving along, delivering the right information. Now, in vestibular dysfunction or some vestibular visual integration dysfunction, you have a slowing of that same information. So that same information is leaving the visual system, but now it's arriving late, which is not happening in real time. It's confusing to the vestibular system. The feedback is delayed, and so you get this poor integration. Now, if you have a visual spatial midline shift, you're not even sending the blue balloons anymore. You're sending red balloons. You're sending completely inaccurate information from the start, and then is also slowed. So that bottom one is going to be the most egregious of the two, because it's not even starting out with the right information. So is you could suffice it to say, by the time that visual information reaches the vestibular system, it's useless, which causes a lot of folks to become more vestibular or proprioceptively dependent. Now, some people still might be visually dependent, but what a lot of folks do then is they'll reach out to walls, they'll walk along a wall with their hand out along the wall, they'll stomp their feet more to get that proprioception, that information, because they can't trust that visual information. I have a video here. You can tell it's old because of the style. I don't know, it's coming back maybe. But this is a pretty dramatic view, but I wanted you to take a look at what it looks like from a kind of a more severe case. This is a man that had something fell on his shoulder and head, and he had a brain injury. So this is him just trying to walk down the hallway. Of course, this is dramatic. If you've had a concussion and had a midline shift, it doesn't mean you can't walk, but it really throws things off. But you can see his sense of where he is in space is totally lost. He's trying to find the floor. He's reaching out with his toes. All right. And then they give him these glasses, and I'm going to skip ahead to where he gets the glasses. And this is him. This is literally 10 seconds later. And he's able to, he's still a little wobbly, but it's a pretty big difference there. This is with prism glasses that help shift the world back to where it's supposed to be. So that midline shift, let's say if it was shifted right, this shifted it back to the left, and now he can understand where he is in the world much more accurately, just from changing the vision. Now, there might be a lot of vestibular issues still here, a lot of proprioceptive issues still here that need other types of work. But from a visual perspective, once we can change the input of that visual system, going back a little bit to at least getting to blue balloons sent to the vestibular instead of red, we can help a lot of that writing process. Let me skip ahead here. So,
Dr. Powers, can I ask a question two questions that just came up?
Sure.
One is, can the visual issues come and go? And the other issue about what you showed with the trucks, I think, is that more a vestibular in the ear system problem, or is it or in the central nervous system, or both? So
It
sorry
could
for throwing both the questions out.
No, that's great. That's great. As far as the first question, visual issues, it kind of depends on which kind of area it's in. I typically see people more with more consistent visual issues, but it's also really heavily dependent on hitting your threshold or not. So
Yeah.
if it's a day when you got great sleep and you didn't really do much the day before and you feel good and there's so many factors to that, then maybe you won't have as many symptoms. But once you hit the threshold, they could all come back. So that I do see quite a bit. I don't see them disappear for like weeks at a time and then come back and recur. But so we could get deeper into that question if the question asker wanted to clarify, or hopefully maybe that answered it.
But yeah, I think that's a great point, because I know for me, those bad days, everything's worse. And people usually, we live a life of normally pushing ourselves. And then with a brain injury, you got to like dial it way back to get things to not hit that threshold over and over. Oh,
yeah.
Yeah.
Yeah. And that can definitely manifest in the visual system, especially because of how energy hungry the visual system is. And so if you hit that threshold and then you try to go do pretty much anything, it's going to involve your visual system. And so that will be affected for sure if you have one or more of these issues. I rarely see any of these issues in isolation. It's usually 5 to 10 things at the same time. So do we want to stay on that? Was there anything else with that, Daniel?
No, I was just trying to throw people's questions at you because the chat's going fast. Yeah.
I love it. Thank you. Yes, I will try to wrap up here soon. So I will not go into all the detail of this. Dr. Toby probably has a better grip on this than I do. I just have a basic understanding of how the vestibular system and the visual system connect intricately. But at least looking at this, you can see that there are a lot of, so these kind of bubbles down at the bottom of each diagram are the nuclei. Those are kind of the hubs from which different connections are made. So here on the top left one here, we see it says right ear. We got those canals that Dr. Taube was talking about. They send connections through lots of different nuclei up to the cranial nerve 3 nuclei, which tells this muscle and this muscle to contract. So if you don't do this at home, but if I am sitting here looking at you and I want to turn my head, my neck, my vestibular system, everything got activated and said, oh, we're turning. At the same exact rate that it's turning, the vestibular system had to tell the muscles around my eyes how much to contract so that the eye muscles would keep my eyes pointed at the screen. So this has to be beautifully calibrated and very precise for your eyes to be where you want them to be when you're in motion. And if we have any kind of dysfunction in any of these nuclei or in the nerves or in the central processing of that visual information, all of a sudden that calibration is going to be off, and that's when we see a lot of dizziness. So there won't be a quiz on this later, but I wanted you to see how deeply connected they are. So up on the top left, that green couch, it's kind of like this map. I was talking about, like, when you look around the room, you're making a map. This is kind of what your visual system does. It's taking in not only color and, oh, what is that? And who gave that to me? And it's the where is it part of the brain. There are 2 different pathways in the brain. We could get into all this fun stuff, but we don't have time. But the where is it piece of information tells us more spatial information and more movement-based information. So we're creating that map constantly. And if you have an inaccurate map, you have multiple systems that are telling you different information. You're constantly recalibrating that map, and it's constantly wrong. It's constantly zapping energy. So you're going to get dizziness and lost balance, fatigue, bumping into things because you don't know where you are. I put an asterisk by decreased cognitive function. This is more something we see when we treat just the visual part. Language and memory and a lot of things improve because this is such a huge energy zap. So that's not a direct correlation, but it's an indirect. We also get nausea because we're unsure of where we are. It's almost like you're getting lurched different places in space and time. And it can cause like a lightheadedness feeling too. On the bottom, those purple diagrams, that's kind of the way that people perceive space is kind of compressed or expanded. And so it kind of changes the way they hold their bodies. So even if you like look in a mirror and walk towards it, if your arm is swinging really big one direction and really tight on the other, your shoulders are off and your hips are twisted and your head's tilted, a lot of people, even without concussion, with those kinds of things, symptoms, have a visual spatial midline shift, but it's way more common after a concussion. Usually they're from some kind of trauma. We assess this with lots of movement, do blind touch testing, spatial midline testing, and then these on the right, this picture is of a trial frame. Yes, they are old school and steampunk, and we still use them because they're so versatile. We look at how prisms, which shift light rather than bending light like glasses, prisms shift the light into the right place. So now our visual system aligns with our proprioceptive system and can put everything where it needs to be and calms down the system, which is really exciting. We can do, the woman here in this picture is wearing really, really thick prisms. We use these as a treatment to kind of change the brain. We put them on and make people do walks and things like that in different positions of prism to kind of expand what they're able to do and the flexibility in that processing. So this is kind of the main area of the brain that is processing spatial information is the parietal, which is up here. And especially the right parietal lobe. And this is something that we really work on in vision therapy to help kind of retrain what that internal map looks like. The last one, I know we don't really have time anymore. Light sensitivity. This is something that affects a lot of people, and there's a lot. I'll probably just blow through this because this could be a whole 2-hour talk, but there's a lot of brain areas that are proposed that have to do with light sensitivity, but we really don't know for sure what happens, why people with concussions are so photosensitive. What we do know from a recent study in 2018 is that the more cocooning you do, dark lenses, dark rooms, the harder it is and the slower the processes of re-exposing yourself to light. So the one thing you can do and one thing you want to avoid is just over cocooning beyond the point where it's helpful for you to be able to function. So the big question is, what are you able to do? And then how much effort does it take to get there? Sometimes people are able to do these things and make it through, but it's just an incredibly energy-rich process, vision is. So always ask that question. And that's it for me. Thank you.